Tamil come to present.
Yes, ma'am.
Ma'am, you may start with the presentation, please.
Ma'am, am I audible? Am I audible and my screen is visible?
You are audible. You can start your session.
Yeah, I'll go with the presentation. Good afternoon, everyone. Myself, Tamilarasi from Karanya University, BTech final year. My project topic is called Attenuation of Quorum Sensing Mediated Virulence in Uropathogen Serratia marcescens by Dialide disulfide, and guided by Dr. I. Silvia Vasantha Pakyavadi, and she is the assistant professor in Department of Biotech in Karunya. And aim and objective of the project is aim of the study is to explore the anti-quorum sensing and anti-biofilm activities of the dialide disulfide, the compound I am using in my project. That is garlic derived. The scientific name is Allium sativum. The objectives are, first, through in vitro studies, I'm going to explore the virulence reduction of dialide disulfide against the Serratia marcescens. The next one is to assess the molecular mechanism of dialide disulfide against Serratia marcescens via in silico analysis by docking with SMAR receptor. The SMAR receptor plays a major role in quorum sensing mechanism. So the problem I am representing here is urinary tract infections. It is very common and most serious infections, and it is affecting 150 million people each year worldwide, and especially it affects children and women. So this complicated, it is categorized into two: uncomplicated and complicated. Complicated infection is more serious than uncomplicated because it affects kidneys directly. And the urinary tract infections are mainly caused by the Serratia marcescens, and it is a gram-negative bacterium, and it belongs to the Enterobacteriaceae family. And there are some virulence factors that are produced by this bacteria are protease, nuclease, lipase, hemolysin, and it also has the ability to swim and swim and to form biofilm. The swimming and swimming motility plays a major role in the first step of biofilm formation. And another important factor is that prodigiosin. It is a red color pigment which is responsible for the color production by Serratia. And it is also responsible for the resistance, and it helps the bacteria to grow well in the adverse conditions. And it also responsible for the resistance shown by the Serratia against antibiotics. So this is the major problem in Serratia marcescens because it is a multi-drug resistance bacteria. So there is a need for there's an alternative need for this thing. So if antibiotic doesn't work, we have to go for another way. That is naturally derived compounds, which I have taken as garlic. So here in this picture, I can clearly explain you how.
Your screen is not moving. Please move your slides.
What, sir?
Your slides are not changing. Please change your slides.
Oh, I'm sorry. From where should I change? From first event, it's not changing?
Yeah, from first of
Is it visible? Is it changing now?
Just continue.
Oh, sorry. I told you the aim and objective, and then introduction urinary tract infections, and then it is caused by Serratia. And then I told you about the antibiotic resistance. And then because of this antibiotic resistance, we are going for an alternative search. And in the second image, is there any problem with the slides again?
Yeah, slides are not changing.
Is it changing now properly?
No.
Okay,
It's
sir.
present. 90
Ah,
plus F5 for presentation.
sir, I'm not able to hear properly what you're saying now. Can I go? Can I continue?
Yeah, please present your slideshow screen. This is your PowerPoint screen.
No, because of that only my slides are not changing properly. So that's why I made it like this. Is it not visible? Ah, yes. Can you please put it
Can you hear
louder?
me?
Ah, yeah. Say.
That where the person is there, right? That we should kindly wait a minute. Kindly wait.
Ma'am, I'm not able to hear you properly. Can you please little louder?
Show the screen, full screen mode. Change your slides to full screen mode. You have an icon down.
Yeah, it's slide
Just
like
click it. Just right clicking it.
slideshow only you're saying, no, ma'am.
Yeah.
So before I was doing this in this thing only, but you said my slides are not changing. Is it okay now? Hello?
They go to your PPT now.
Yes, ma'am.
Not in this way. Go to your PPT.
Ah,
And in your PPT, make it a full screen. You have
I
to see the cursor in that particular place. You can clear that icon. So just click it in your presentation.
okay, ma'am. Wait. Where exactly?
You have a PPT right at the back.
Reading view.
Not in the presenting screen. In your market, you have a PPT in your desktop, right? Was okay?
Am I going?
Can you see the cursor?
Oh, wait, ma'am.
Can you see a cursor?
Mm, can you tell me again where to go?
Can you understand, Tamil?
Show? Is it slideshow?
PowerPoint கீழே வந்துட்டு உங்களுக்கு full slideshow காமிக்கிறது ஒரு icon இருக்குல்லடா.
அதான் கீழே press பண்ணி தான் ஃபர்ஸ்ட் ப்ரெசென்ட் பண்ணேன்.
ஆ.
அப்பதான் ஸ்லைட் சேஞ்ச் ஆகலன்னு சொன்னாங்க. அதனால ஸ்கிரீன் இப்படி மாத்திட்டேன்.
You can continue. You can continue with the same slide.
Huh?
You can continue with the same slide.
Okay. Sorry for the inconvenience. I'll continue.
Yeah, continue.
Thank you. Yeah, here because of this biofilm formation, antibiotics are not able to act. And if we disperse this biofilm formation, antibiotics will be able to go through and then they can heal. This is the biofilm formation role in antibiotic resistance. And I'll tell you about quorum sensing. This biofilm formation is happening because of quorum sensing. Quorum sensing is nothing but cell to cell communication. Here, in this picture, you can see an individual cell. It cannot act by itself against antibiotics. But when it is in group, they can act well and they can create this antibiotic resistance. So this is called quorum sensing, and this is the advantage of every bacterial cells. And this quorum sensing helps in bacteria in every ways. So my main goal of the project is to inhibit the quorum sensing instead of killing it. I'm not going to kill the bacteria. I'm just going to inhibit the quorum sensing mechanism, and it is going to stop the virulence factors coming out from the bacteria. And the natural source I've chosen is garlic, because you all know already that garlic is the traditional medicine that have been used for a long time in our southern parts of India. And plant derived compounds are always not a bad ones, and it's a good one, and it is a natural resources. It does not cause any kind of side effects to the people. And from that allium sativum, I have chosen one compound, diallyl disulfide, that's simply called DATS. This compound is most present in garlic, which have the content of 20 to 30%. And this is the workflow of my project: in vitro studies and in silico studies. In vitro studies, I've done 4 assays, and I've also found out the MIC value of the DATS, and in silico studies to give a confirmation about how it is going to work. And this is the pure culture of Serratia marcescens, and this is the strain number, and this is the overnight culture of Serratia. You can see the color. This color is because of the prodigiosin pigment, I've already told you. And this prodigiosin pigment production varies according to the temperature variance. And I'll go directly to the procedure. Determination of MIC. MIC is nothing but least concentration that inhibits the bacterial growth. And this is the methodology I've followed. And here in the results, I've found out that the 60 mg per ml is the minimum concentration of DATS, which could inhibit the growth of bacteria. But my project is not about inhibit the bacteria. It's about reducing the quorum sensing potential. So below that 60, I've chosen 30 mg per ml and 50 mg per ml as my concentrations to use for the assays I'm performing. And here you can see the color difference between all those things. In the first image, it shows CC, that is called and the 30-60, it contains 30 mg per ml, 60 mg per ml concentration of compound in test tubes. So that's why it reduced the prodigiosin pigment reduction. And then in 60, you can see the clear solution like blank. So in this 60 mg per ml, it has reduced the growth of Serratia completely. So instead of that, I'm taking 30 and 15 mg per ml as my sub MIC values. And the next, I've performed assays to give a confirmation that my compound is working against bacteria. So next assay is prodigiosin assay. I've told you already the prodigiosin is very important in bacteria, in Serratia bacteria. So in this, I've done this a method by following this method. In this image, you can see that it has reduced the color. And at 30 mg per ml, it has reduced the pigmentation by 70%. And in quantitative analysis also, you can see the graph that the reduction of prodigiosin pigment as the concentration increases. Again, this confirms that this compound is working against bacteria. And I've done the swimming assay. I've done the swimming assay using this method. In qualitative analysis, you can see that there are 2 controls I've kept. In that, you are able to see without compound, it actually can swim well. But with compound, it can't swim. So here the quantitative analysis results. In control, it grown till the end of Petri plates, that is 7 centimeter. But in 50 mg per ml and 30 mg per ml, the migration zone is very less compared to the control. So again, this compound is working against bacteria. And then swarming assay. I've done the swarming assay using this method, which is already described in the flowchart form. And here in qualitative analysis, you can see the swarming zone. And here in treated Petri plates, you can see that only you can't see anything because there is only one round at center. The compound did not allow the bacteria to swim well. And you can see the results here. Control, it is 4 centimeter, 50 mg per ml and 30 mg per ml. It is 1 centimeter and 0.8 centimeter. And again, biofilm biomass quantification. Here, I've done the assay by following this method, which is again in the flowchart form. And here, qualitative analysis, you can see the difference between four these images. The culture, the biofilm ring is shown well, and then 7.5 mg per ml concentration, you can see a little reduction, and then 15 mg per ml, you can see again a little reduction, and at 30, you can see almost the biofilm formation has gone. And the quantitative analysis, you can see the reduction of biofilm formation in the graph. And then already I told you I'm not concentrating on the killing of bacterial cells. So here in this graph, it is very well explained. At subbimaces, 7.5 mg per ml, 15 mg per ml, 30 mg per ml, diallyl disulfide did not inhibit the growth of Serratia marcescens, which confirms the QS activity of DAPs. That is, the diallyl disulfide is not working as an active antibacterial agent; it is working as an anti-quorum sensing agent. And then in silico studies also confirmed that it can interfere the binding of natural ligand with SMAR receptor. Simply, I'll just tell you this natural ligand C4HSL is binding with the SMAR receptor. That's how the quorum sensing is happening. So the diallyl disulfide and the natural ligand C4HSL almost have the same similar structure, same morphological characters. That's why this compound is able to act well, and it can interfere the binding of C4HSL and SMAR receptor. And in instead of C4HSL, the DATs will go and bind with a SMAR receptor, and it will not increase the quorum sensing mechanism. Instead, it will decrease the activity of quorum sensing, and it will interfere all the activities, and it will stop all the virulence factors, and it will not inhibit the growth. It will inhibit the quorum sensing mechanism. In this picture, first tabular column, you can see there are 6 attachments between natural ligand and SMAR receptor, and the highest affinity score is minus 5.8. And then again, I've done molecular docking between diallyl disulfide and SMAR receptor. In this also, you can see there are 5 attachments between diallyl disulfide and SMAR receptor, and the highest affinity score is minus 4.2. From this, you can say diallyl disulfide acts as a competitive inhibitor and does not allow the natural ligand to bind with the SMAR receptor. It prevents the binding, and it is acting as an anti-quorum sensing agent. That's all. Thank you.
Thank you, Dr. Rachina.
So, any doubts or any questions from?
So, if you have any questions, you can ask. Yeah, okay, thank you, ma'am.
Thank you, sir.
We call upon Roshni. Roshni, are you ready with your presentation?
Yes, sir.
Yeah, okay, ma'am, you can start presenting your screen.
Is my screen visible? Good afternoon, everyone. So my topic is evaluation of amla, haldi, and methi against targets alpha amylase and DPP4, that is dipeptidyl peptidase 4, for management of type 2 diabetes mellitus. It's an in silico study.
Roshni, can you hear me?
Yes, ma'am.
Your slides are not visible. Can you please present your slides?
Yes, ma'am. Ma'am, now the slides are visible.
Yeah, can you make it into landscape, like in full screen?
Ma'am, are my slides visible now?
Turn your phone into landscape mode so that it will be more clearer.
Ma'am, now is my slide visible?
Share your slides, Rashmi. Can you please share your slides?
Ma'am, I have shared.
Okay, shall we present your slides for you? Is it okay if we present, share your slides? You have your slides, right?
Yes, ma'am. Yes, ma'am.
Okay, once again. Rashmi, did you make your slides?
Um, Ma'am, you're not audible.
Roshni, did you share your slides with us?
Yes, ma'am.
But it's not there in the mail ID.
Ma'am, is now visible?
Can you please email your slides to us?
Okay, ma'am.
Thank you. Hey, you also busy getting shared here. Okay, Roshni, you can start presenting. Roshni, can present your slide. Switch on your mic, Roshni, you're not audible.
Ma'am, is my screen visible now?
And now you're audible.
Ma'am, and my screen is visible?
Now your screen is visible.
Okay. So good afternoon, everyone. My topic is evaluation of amla, haldi, and methi against targets alpha and DPP4, that's dipeptidyl peptide 4 for management of type 2 diabetes mellitus and 8 glycoside. So the abstract is alpha amylase and four are important targets for the management of diabetes mellitus. Inhibition of this enzyme decreases post-corrective hyperglycemia. The goal of this research is to examine phytocompounds of curcuma longa, Helianthus america, and Chromium gracium, which are commonly type which have the ability to inhibit alpha and DPP4 protein, which upregulated. Biomimetic tools and techniques such as molecular docking have been utilized to narrow the plant and bioactive compounds. So first, what is diabetes mellitus? Diabetes mellitus is a serious, complex, multifunctional disorder characterized by hyperglycemia, that is very high level of blood glucose, and glucose intolerance, either due to the relative deficiency in insulin secretion or impaired effectiveness of insulin action to enhance the uptake of glucose. So there are classification of diabetes. The diabetes mellitus is classified into three main types, that is type 1, type 2, and gestational. Type 1 diabetes, type 1 diabetes is an autoimmune reaction. This reaction destroys the cell in the pancreas that makes insulin, called as beta cell. This process can go on for months or year before any symptoms appears, and it is also called as insulin dependent type 1 diabetes mellitus. Type 2 diabetes mellitus is an adult or onset diabetes. Here, the cells will become resistant towards the insulin. The pancreas will produce insulin, but our cells will become resistant. So there is no uptake of glucose. By using traditional herbs, we are going to cure the diabetes. Like we have the modern medicines, which are the first line of defense in type 2 diabetes mellitus, that are acarbose and sitagliptin. Though these are the chemical process, we have side effects of using this medicine. So we are going to, in this research, we are going to see the traditional herbs with the standard drugs. So by the research and the literature's study, we have come to know that three plants, that is curcuma longa, Helianthus amelica, and Trigonella fomium gracium, are the three major plants which are known to be anti-diabetic. So the in silico study have been done. For that, we have chosen first the plants have been chosen as I have told by literature search. Then the screening of phytocompounds of all the three plants, their PubChem IDs and canonical smiles. This in silico data forms a base in understanding the interaction between the standard and the phytocompounds which have been screened from the various plants. And further, we need to study the in vitro or the animal model to get more knowledge about the herb drug interaction. So the proteins are alpha amylase and DPP4. Alpha amylase are the digestive that are the carbohydrates and increases the postprandial glucose level in diabetic patients. Inhibiting the activities of these two enzymes can control the postprandial hyperglycemia and reduces the diabetes. And this DPP4, which regulates the metabolism, since it can cleave and inactive the peptides such as glycogen like peptides. They also inhibits the activity in peripheral plasma, which prevents the inactivation of the incretin hormone glycogen like peptides in peripheral this plasma. Molecular docking was done. There are three steps, that is ligand preparation, identification and preparation of proteins, and its active site determination, and ligand target docking. So ligand preparation, the PDB dataset was used to extract the structure of the ligand attached to the various receptors of type 2 diabetes mellitus. In this phase, we have listed around 660 phytocompounds from all the three plants. Those were anti-diabetic from, and then their PubChem IDs were copied and the canonical smiles, and the STF format of these were converted into the PDB-QT format using that was used it was an requirement of for docking process. Then identification and preparation of protein target. The protein molecules for type 2 diabetes mellitus was identified through the literature, and they were selected as for the standard drug used for type 2 diabetes mellitus. In this study, as I have mentioned, two drugs we have used, that is acarbose and sitagliptin. Then the active site and the grid preparation was done. The selected plant protein molecule have been assessed for their active site based on the literature, and the active site was uploaded by using the PrankWeb website, and the grid was formed. We can go for blank docking or else the grid docking. Uh, yeah, as we performed the blank ducking, the grid for the protein was set at the maximum, and screening of both the standards as well as the phytocompounds were there, and the exhaustiveness was kept under 25. So the standard and virtual screening by molecular docking, the standard and the phytocompounds that were selected for virtual screening were screened using the blind docking method for both the standard drug and the phytocompounds. And the pockets for the protein alpha amylase and DPP4 uh, was predicted, and their active sites were matched with the phytocompounds of the various plants. So this is the protein and ligand interaction. Alpha amylase, their uniprot ID, PDB ID, and the ligand acarbose. DPP4, uniprot ID, PDB ID, and the standard drug, that is sitagliptin. As acarbose is an inhibitor of alpha amylase and sitagliptin is an inhibitor of DPP4. Standard and active site pockets for alpha amylase. Uh, the binding energy was minus 8.3 and DPP4 was minus 8.2. The active site pockets matched with the phytocompound residue was 196, and for DPP4 it was 53. So this is the 3D image of standard protein and our phytocompound, and this is a 2D image of this protein and standard. So results predicted the major phytocompounds of all these three plants, that is Curcuma longa, Helianthus umbilica, and Trigonella fomium gracium, was uh, were binding at the same site as that of the standard, that is acarbose and sitagliptin. The phytocompounds of curcumin was dis-methoxycurcumin and curcumin from the Curcuma longa, malic acid from Helianthus umbilica, and phenylgreekine and quercetin 3,7-diglucoside from Trigonella fomium gracium, having the residues in common with the least binding energy, and were predicted to be competitive inhibitor of both alpha amylase and DPP4. Thank you.
Thank you, Roshni. Thank you for your wonderful presentation.
Any doubts?
Now, third, just a clarification. What's the future of your work, Roshni? Roshni, can you hear me? Sorry for the inconvenience. Thank you, Roshni. Thank you for your presentation. Now I would like to call upon the next presenter.
Kumar MP.
I request Uday Kumar and team to present your work. I request Uday Kumar and team to present your presentation.
Okay, ma'am. I'll present my work now. Ma'am, is my
Yes, it is visible.
okay, ma'am.
Kindly
Let me
present
check.
your slides.
Good evening, one and all present here. My name is Uday Kumar. I'm just studying in College of I'm going to present the study of considerable nitrate reducing bacteria in the Arabian Sea in the insight from oxygen minimum zones and non-oxygen minimum zones. In this study, we analyze compare the microbiome in the oxygen minimum zones and non-oxygen minimum zones. So, when we start to say about Arabian Sea, this contains the maximum amount of oxygen, the oxygen minimum zones, where the oxygen level is 20 mole per liter. It is so low when compared to the normal zone levels. It is because of mainly because of the northeast monsoon and southwest monsoons. During this time, winds will agitate and form eddies. Eddies means the circulation of water which forms as sludgy compositions and form an buoyant region. In this areas, the microbiome will utilize the nitrate as a and increase their nitrate as a main source and increase their nitrate. So they go under canonical denitrification and ammonium nitrate reducing method to undergo this biochemical reaction and utilizing it. They convert nitrate into nitrite. This increase will fix the remineralization process in the nitrogen cycle, which is mainly responsible for the monsoon and monsoon season and also the lower level nutrient things. So in this study, our objective is to culture and isolate the heteromorphic extremophiles, analyze of the physiochemical variables on the OMZ and non-OMZ region, and meryl chlorophyll analysis, chlorophyll A analysis, and TOC analysis. And we have also done nitrate assay from understanding for understanding the biochemical pathways. In later, we are moving towards the phylogenetics and biomarker analysis. In physiochemical variables, we analyze pH, salinity, DOD, turbidity, temperature, and content in this OMZ and non-OMZ regions. This table shows where we collected two samples in OMZ and non-OMZ regions in different variables like from 55, 110 meters and 210 meters and 300 meters. And we analyzed the chlorophyll and analyzed the bacterial abundance and analyzed how much they are reducing the nitrate. These are the physical parameters. Here you can see that temperature drops in, when compared to the depth, lowering the temperature. Fluorescence. This fluorescence is mainly to detect how much chlorophyll is. We are seeing only chlorophyll A because the cytoplasm contains more amount of chlorophyll A, which reproduced based on the photosynthesis. So that's why the fluorescence we are measuring. We are measuring the fluorescence. One is to identify how much cytoplasm. Two is to analyze how much sunlight is getting into that. Here you can see that the peaks in the OMZ region. That orange color is OMZ region and blue color is non-OMZ region. In that, you can see that under the depth of 150, you can see secondary chlorophyll maximum. It is because when there is a high amount of chlorophyll, when there is high amount of phytoplankton, utilize the organic compound and without oxygen which is present on the surface above the thermoplane and which develop the OMZ region. So in later depths of 110 and you can see a small amount of, that is there is only low amount of oxygen present in that region. And you can see the dissolved oxygen also, it's getting lower and lower when compared to non-OMZ region. This is primarily because of this seasonal monsoonal winds, next phytoplankton community, then also by the bacteria, which is raising the nitrate and producing increased amount of nitrate. This chlorophyll analysis. Here you can see two charts that 11, that green line is OMZ region and blue line is non-OMZ region. Here you can see large amount of, there is no significant amount of change in chlorophyll because we have because after the southwest monsoon, southwest and northeast monsoon, because like after this only the OMZ is developed in the Arabian Sea, which could clearly gives us the values of chlorophyll and amount of bacterial abundance which is present in that. In that, we could able to isolate about 65 bacteria morphotypes and 21 fungal morphotypes. Here you can see the CFU units of each place, and the pictures which have shown have different varieties of bacteria which I've chosen. And we also study the diversity indicators within past software. And here you can see that the values shown and more amount of dominance in this 55 meters and 110 meter. We follow dominance, Simpson and Shannon index for the diversity concepts. And we also did this nitrate reduction test through Grease test. In this, we add these two reagents like reagent A and reagent B, which contain sulfonylamine and alpha-naphthalene, which will reduce the external nitrate which is remaining in the samples after the bacteria is utilizing it. In that, you can clearly see that first 10 samples are OMZ region and next 20 samples are non-OMZ region. In that, the first 10 samples which are OMZ shows positive result for nitrate, and other remaining when in compared to 20, there is only 9 cultures are showing the nitrate reduction assay. This comparatively 50% lower that non-OMZ region have a 50% lower bacterial community which reduces the nitrogen because they are mostly other substrate utilizing bacteria. Thus, coming to summary, we could see that both surface sea surface temperature is 27 plus or minus 1, and the dissolved oxygen in the surface is 4 and relatively it drops after reaching this thermocline surface, and it will go when it reaching the OMZ region, it is reducing more and more. And the chlorophyll control also trivially changed because that we are not analyzing during the southwest monsoon, we are analyzing it after the southwest monsoon and OMZ region which is formed. And we could like after this serious test, we could find the 30 unique morphotypes which are present. And now we are given the samples for NGS data, NGS and other things. In that we have found that more bacteria which we have are proteome bacteria. And now we gave the sample for sequencing. Soon we'll get. Why I have done this comparative study and present here, because this environmental study will give an overview of OMZ region and how these bacterial community influencing the organic substance which are present in the ocean, and also it helps some like industries to understand the OMZ region and do something regarding this. This is a natural process, but in sometimes the fish and other things which are present in that regions are affected more during the seasons. So this comparative study will help these researchers like to understand more about the OMZ region. And these are the references which I'm analyzed for this. And thank you, ma'am.
Thank you, Dekumar. That was a nice presentation. The session is open for discussion.
Ma'am?
Yes?
Ma'am, thank you. I'm done with my presentation. Is there any question?
Is there any questions? Any clarifications? Thank you. That was a nice presentation, Dekumar. Thank you. I would like to call upon Subhashri for the next presentation.
Ma'am, I couldn't hear you properly. Can you speak?
Can you hear me?
Yes, ma'am.
Thank you, Dekumar. That was a nice piece of presentation. Thank you.
Thank you, ma'am. Thank you.
I would like to call upon Subhashri for the next presentation. Subhashri?
Yes, ma'am.
Please, you can present your slides.
Okay, ma'am. Good afternoon to all. Hello,
You're audible, you can present.
ma'am. I'm getting my voice repeated, ma'am.
Now you can present your presentation.
Okay, ma'am. Good afternoon to all. My topic is standardization of screening technique using tissue culture plants. Potato cyst nematode and root knot nematode, the PCN and RKN. Here it is based upon the agricultural research, my project. Here, aim of my project is to screening technique using the tissue culture plants. First of all, the potato cyst nematode and root knot nematode. Potato cyst nematode is a pathogen which is present in the soil where the potato is cultivated. It absorbs the nutrients and it reduces the growth of the potato. A potato cyst nematode is a cyst. It lays a cyst which contains the n number of eggs, and root knot nematodes is lays eggs like egg masses. Uh, Both will be is a pathogen which affects the potato. My objective is cyst was collected and they are surface sterilized by using the chemical reagents. The chemical reagents which is kill the pathogens and microorganism which is also present along which is present along with the cyst and egg masses. The media was prepared and the cyst and egg masses are inoculated into the media and along with the cultivars like the potato varieties which is resistant and susceptible to the potato cyst nematodes and RKN. Here I'm using the media is B5 media. First one is the potato cyst nematodes, and below it is the root knot nematode. Potato cyst nematodes is lays egg after outside the host, and root knot nematodes lays eggs along in the root. First, I've isolated the PCN and RKN from the soil and root samples, and these are the PCN, these are the RKN, which is separated from by using the Fenwick's method. Then surface sterilization was done using the three chemicals, which is mercury chloride and sodium hypochlorite and streptomycin sulfate. Then these chemicals is treated like 23 treatments, used like a 23 treatments. Then the use of media preparation. B5 medium is suitable for the potato plant cultivars because it is a consist of 5.8 pH level, and they consist of macronutrients like potassium nitrate, calcium chloride, magnesium sulfate, ammonium sulfate, and sodium hydrogen phosphate. Then micronutrients and iron stock and organic stock and agar and sucrose. Inoculation is a technique I used to inoculate cyst and RKN using the dry method, using the inoculation loop. Dry method was drying the completely the drying of cyst and egg masses. In case we inoculated it along with the water, it can make causes the contamination, carries the contamination. And the observation after the 23 surface chemical sterilization, after the 11 days I got contaminated, and 7 treatments I have achieved for the not contaminated until the 1 month. The treatments after the 21 days, treatment 3 and treatment 9 and treatment 15, these are the treatment along with the concentration of mercury chloride, which is different time incubation. They have succeeded for surface sterilization of the PCN and RKN roots, eggs. And the cultivars I used here for the cultivation is Kufri Girdari and Kufri Jyoti, Kufri Sahitri, and Kufri Swarna, because the Kufri Girdari and Jyoti is susceptible to the PCN and RKN. Sahitri and Swarna is resistant to the PCN and RKN. I have done the in vitro study and in vivo study. In vivo study, I'm going to comparative like one inoculating the one cyst for the for per pot in the sterile soil. In vitro, also inoculating the one egg masses and one cyst uh, in the media. In this study, I'm going to tell about the one inoculating the one cyst. It cannot have the hatching ability in vivo study, but I'm going to succeed in the in vitro study. Then the 45 days of for the RKN hatching study will be observed, and the 60 days of PCN hatching study will be observed. Then the treatment like 3 and 15 had no contamination after the until the 60 days. Then I analyzed the cross study. This is the pot analysis after the 19th day. Then hatching test is for the cross check of the hatching study is done using the potato root diffusates, which is soaked for the girdari plant 15 to 10 20 days old kufri girdari plant was taken and washed in the root and soaked it with along with the distilled water for the overnight incubation, and next day I filtered double filtered the water and used it for the hatching test study for the potato cyst nematodes. Then the juveniles have hatched. Then I counted the juveniles according to the treatments. Then on that result I have succeed like treatments and hatching incubation has increases, then it controlling the hatching ability. It results that my surface sterilizations is also killing along with the eggs in the cyst. So then egg viability test also have taken for that. In that Meldola dye blue test is dye is used for this study. In this study, I have soaked the PCN in the Meldola dye for 7 days, and after the incubation, alive will be determined as a colorless, and dead is determined in a colored, like blue in color. Here, this is the study of the egg viability test. In this, I have found dead eggs also. So this is a means that my mercury chloride killing the egg inside the cyst. So I have done another study like for in vitro soil because the surface sterilization is not succeeded. So we have used the germination paper and water and pepper for the study. I have made the foldings, and this is the 2 into 2 foldings have done for the inoculation of plant. Then it is a length of 30 centimeter and breadth of 15 centimeter. Thermocol I have used for the support of the paper to stand vertically. In this, I have used Sayadri and Girdari plant, one resistant and one susceptible plant for the study. And I inoculated the juveniles into the root. So I have observed the cyst and egg masses as produced, as developed. That is, the hatching is capable in this germination paper is succeeded. Thank you.
Thank you.
Yes, ma'am.
That was a good presentation. This session is open for discussion. Any queries? So we should have one clarification. I need a clarification.
Okay, ma'am.
You said you have to 100 ppm for 1 hour, right? With mercury sterilization with mercury, 100 ppm for 1 hour, right?
Ma'am, I can't get you, ma'am.
You have used 100 ppm for 1 hour for sterilization, surface sterilization, right?
Yes, ma'am.
So what the good toxin?
Ma'am, I'm telling you is shortly, ma'am. Like my my study is about in vitro study, but like comparatively study for like one cyst in pot, like in a soil, it unable to hatch, ma'am. So it can't replicate itself. So I have used one cyst, but it can be succeeded in vitro study. That is a main goal of this project. So in this, in because soil also consists of the microorganisms. It will be along with the outside of the body of the cyst. So it can carry the contaminated in the in vitro study using the media. So we have alternatively we have used the germination paper. In this, there is a no there will be a growth of bacteria, but that can't inhibit the hatching ability of the potato cyst and root knot nematode in germination paper, ma'am. That is this. But the chemical sterilization has not succeeded because the mercury chloride is a toxic chemical. For we are using it for the plant tissue culture for sterilizing the egg plants. But in this, it also affecting the root knot nematodes eggs and potato cyst nematodes eggs, ma'am.
Because I was wondering like mercury chloride is mercury is toxic, right? Basically, so it's not volatile, that's why I asked you. And your inoculation, how much of eggs do you inoculate and how do you calculate the numbers?
Ma'am?
For inoculation, you have inoculated your medium, right? Initially, you said you have inoculated
Ma'am?
your medium, right?
Yes, ma'am.
How do you calculate the numbers, these numbers?
Cyst, like in a microscope study only, ma'am, like collecting one cyst for a one cyst is inoculated in the one test tube, ma'am.
So your question calculate how much of medium?
Ma'am?
Like for what number of cysts were inoculated? What's the question? How much volume?
Ma'am, one cyst per one tube, ma'am.
Sorry?
One cyst in one test tube, ma'am.
No, how much ml of medium you have inoculated? One cyst.
Ma'am, I didn't get you.
Okay, fine. Thank you. That was a good presentation, Sinushi. Thank you.
Thank you, ma'am.
I kindly request Selin Maria to present.
Ma'am, whether my slide is visible, ma'am?
Yes, it is visible.
Good afternoon to honorable volunteer. My topic is about screening of potato cultivars against root knot nematode. And my objective of the study is screening of cultivars against root knot nematode and acid fusion staining techniques and collection of dead potato cyst nematode by cyst extractions and root galls in root knot nematode. Introduction of root knot nematode. A root knot nematode were placed in the subfamily of Melodonisa, which is ranked as the most economically damaging crop of plant pasting nematode. Root knot nematode attack a wide variety of plants and become serious pests in the home garden. They are microscopic roundworms that live in a soil and plant roots. Root knot nematode are associated mainly with light soils, but most damage is caused under glass, particularly in hot conditions. Root knot nematode larvae infects the plant roots, causing the development of root knot galls. The samples were came from Gujarat. I planted 17 pots. The variety of potato I planted is red cherry rosette. Per pot, 3.5 kg soil is added. The soil is terrace soil and a farmyard manure is added a 3:1 ratio. Screening done after 60 days. The initial population of cysts in terrace soil is 7. For farmers in Gujarat, yellow is a flavor of season since it is red. And this type of potato is simple handle and weighs nearly twice much as other varieties. The color tuber skin color is red and eye color also red. A primary tuber flesh color is light yellow and shape is round. Tuber skin structure is rough and smooth to intermediate. This is extraction. FTW Jones, who found a cyst in the year 1961, I done cyst extraction by Fenwick method. And the protocol is to mix the soil, weight 100 grams of soil, and wash the soil particles. Heavy particles are retained in the soil feeder, and large particles and cyst flows in the upper surface. The cyst are collected in two sieves. In first sieve, the mesh size is 30, and second sieve is 60 mesh. In lower sieve, the cyst is collected. And these are the treatment of that 17 pots. In T1 infected tubers, washing water added, and T2 infected tubers with prout, T3 infected tubers with PTM, and T4 roted tubers of LR, and T5 infected tubers with total ones, and T6 untreated seeds. If untreated seeds means the potato is not washed with boric acid. And acid change. After the 60 days, I took the roots for acid fusion staining. 25 ml of 0.2 gram of acid fusion staining, and 10 ml of stock solution is taken, and 90 ml of distilled water is added, and it kept in for boiling. And the roots are first washed with sodium hypochlorite and inoculated in acid fusion for 30 seconds, and remove it and washed it with distilled water for more stain if it occurs. And these are the treatments. T1 I took 2 plates, T2 2 plates, and T3 5 plates. T4, T5, T6 I took one plate. And these treatments, and these are average of my treatment. Actually, my main concept is that in Gujarat, the farmers are taking roasted potatoes and washing water. Again, they are using in the field. So the root knot nematode is it affects the next generation. Thank you.
Thank you. Any queries? What's your future work, Sylvie?
Ma'am? Ma'am?
What's the future plan of your work? Which perspective? Can you hear me? Can you hear me? Thank you, sir. Thank you, sir.
Thank you, ma'am.
I request Leena Sharan to present your slides.
Okay, ma'am, I'll present it now.
Leena,
Is my slide visible, ma'am?
Yes, it is visible. Please present.
So the topic is marine algae incorporated ointment for wound healing. So wound healing is process includes inflammation, suppression, cell proliferation, and collagen tissue contraction. Here, the wound healing process is complicated, but natural process helps in remodeling the tissues and tissue formation. Burns, incision, excision are some types of wounds that can happen. So, an effective and local wound healing treatment is always in demand. Our my active source of bioactive sources, Cuppophycus alvarezi, marine red algae, bioactive compound reservoir. Not many studies about Cuppophycus on wound healing has been reported. This Cuppophycus are known for good antioxidant activity. Wound healing properties need antioxidant activity. So we coupled this seaweed for wound healing property. So the methodology include collection of marine algae, extract preparation, and preparation of ointment with algal extracts, in vivo studies, biochemical and histopathological studies. So we prepared a simple ointment base to which 5% of Cuppophycus alvarezi
You know,
extract has been
sorry to
added.
interrupt, Tina, but
Cup-
please change your slides.
yes.
The slides are not, your slides are not changing. Yeah, now it's fine.
Now,
Yeah, it is fine now.
okay. So from which slide should I continue?
You can continue with your presentation.
So from
first.
this, I'll continue from methodology.
Okay, fine.
So the methodology includes collection of marine algae, extract preparation of ointments with algal extracts, in vivo study, biochemical and histopathological studies. So here we prepare Cuppophycus alvarezi incorporated ointment. The ointment base was prepared, to which 5% of extract is added. To another group we added 10% of extract, and to another 15% of Cuppophycus alvarezi extract. Cuppophycus alvarezi extract was prepared by aqueous methanol preparation. So 95% methanol and 5% distilled water has been used for extraction process. Cold extraction process has been used for extraction. So we took cis albino mice for study purpose, in vivo study purpose. We induced excision wound model. We did excision wound model, to which we applied the ointments for 16. We had a treatment period of 16 days. So the results showed better results in the ointment, which had 15% Cuppophycus alvarezi extract. So the period of epithelialization was in the order of 15% Cuppophycus alvarezi extract. We also compared the prepared ointment with commercially available ointment too. So we got better results comparing to the commercial ointment. So only the untreated group. That's untreated is we didn't apply any ointment or any treatment was given to that group. That group we considered it as ointment untreated group. So only the untreated group showed the longest time for period of epithelialization. So same results were found in percentage of wound closure and area of wound. Body weight of the mice were studied during the treatment period. It was observed only during the time of wound. The mice was less in range. The animals which were selected for study was in the range of 26 to 36 gram of weight. But when the wound was induced, the weight of the mice started reducing. But the mice which had treatment, had ointment for it, there was not much difference in weight, and they maintained the same weight till the end of the treatment. Only the less weight was observed only in the untreated and ointment based groups. So these are the photo evidence of skin. You can see untreated mice had wound till the 12th day, and on the 16th day we had even the dents of it. Control also had the dents. Standard ointment also had the dents. Whereas Kappa ficus alvarezi, 5% of Kappa ficus alvarezi ointment had dents. 10% you can see there's a regrowth of hair happened there. In 15% also regrowth of hair happened. The histopathology studies also showed granulation in 5%, 10%, and 15% of Kappa ficus alvarezi showed there was no inflammation. Whereas in 5% there you can see granulation tissues. Inflammation filtrates also, but in 15% you couldn't find. It showed normal epithelial cells, and in 10% reepithelialization was also found. So the results showed the prepared Kappa alvarezi incorporated ointment was found to be stable, thicker, opaque, and maintain its structure even at room temperature. Epithelialization percentage of wound closure found to be higher in 10% and 15% Kappa alvarezi incorporated ointment. As the wound healed, the body weight monitoring showed a rise in temperature. We also studied the liver and renal parameters, which also showed normal levels in 10 and 15% of Kappa ficus alvarezi incorporated ointment. The conclusion can be based on the results of the present investigation. It can be concluded that Kappa ficus alvarezi incorporated ointment versus wound healing property. Further isolation of bioactive compounds responsible for the activity has to be taken up, which may possess cost-effective or less or no adverse side effects. Also, can also focus on other mode of drug delivery. For example, we had ointment here. We can also focus on preparing hydrogels or nano particle incorporated hydrogels for wound healing with Kappa ficus alvarezi as bioactive component. And thank you.
Thank you, ma'am. That was a nice presentation, nice piece of work. The session is open for discussion. Any queries? Rina, I just have one clarification.
Okay.
You have seen closer, right? You have seen histopath for inflammation.
Yes, ma'am. My voice is not very audible.
Can you hear me now?
Yes, ma'am.
You have seen inflammation and histopath alone, right? Have you looked for any other molecular techniques or any other molecules?
Ma'am, sorry, I couldn't hear you properly. Can you? Voice is not clear actually.
Can you hear me now?
Okay, yes, ma'am, but some disturbance. If able to post question on chat or something like that.
One second.
Can you hear me now?
Yes, ma'am. Yes, ma'am.
Okay, you have looked into some inflammatory molecules only in histopath. Have you looked into any molecules at all?
Inflammatory cytokines you are asking,
Yeah,
ma'am.
yeah, either cytokines or any proteins.
Yes, ma'am. We also have done inflammatory cytokines as well as MTS collagen expression also, ma'am.
Okay, so how was it?
The same results, ma'am. 15% and 10% of Kappa ficus alvarezi showed better results. In collagen expression also we got better results, ma'am.
Okay,
Inflammatory cytokines has been reduced in these groups also.
okay, what were the panel of cytokines that you have been looking for?
TNF alpha and interleukin, ma'am.
TNF alpha. Okay, thank you, ma. That was a nice presentation. Thank you.
Thank you.
Thank you every online participants who have joined in this Google meeting. The session ends now.
The session link will be posted in your respective conference group WhatsApp groups. The valedictory session will be on time. The time will be noted, informed in your WhatsApp group. Please join during the session. Thank you. Have a nice day. Arun sir.
Now, I'd like to call Nandini for a paper presentation.
Good afternoon to all of our present here, respected faculties and all my dear friends. Today myself, I am Nandini from Department of Biotechnology of NCBESD. I am here to present my research work that is titled Study on Drosophila melanogaster with respect to its sleep and wake behavior and dietary modifications. I would like to give a brief and small introduction to what my research has been about. In small, I connected three topics. First is that of Drosophila melanogaster that has been my very much interest as a genetics student, and then a normal phenomenon of sleep, which we see how easily we can just take a hard nap can give us so much energy. Sleep, and the third I connected was that of sleep comes with coffee. We take coffee to like whenever we feel low. So connecting these 3 points, I conducted this study. Next. So Drosophila melanogaster is actually a fruit fly which we often see in a rotting fruit or in vegetable, and it is one of the very important model organisms in genetics as due to its short life cycle, and it has small rearing time, less inexpensive as compared to other model organisms that we can see. Next. So here we connected, according to some theoretical studies, it says that sleep regulation is very important for insects as when we are offline, that is the sleeping mode, when we are offline, it also has to do the foraging activity as well as it has to have it's all the metabolic processes that the foraging, mating activities going on. So to strike a perfect balance, it is very important. So in sleep regulation, it comes with 2 main processes that give the sleep. That is the circadian clock and the sleep homeostasis. Circadian clock is nothing but it is said that the 24 hours day and night cycle that regulates the human being. And sleep homeostasis is nothing but if we hadn't have enough of sleep, the blood pressure that is generated in the body, which is called as sleep pressure, and which when we compensate that sleep, it is known as the sleep rebound. So flies also exhibit very much similarity to that of hallmark of the mammalian sleep. So we took easily available Drosophila for this study. Next. So when we are sleepy, of course, sleep is associated with when we are having a sleepy or when we are feel of tired, we often take coffee. That is the basic thing. So caffeine is nothing but it is a secondary metabolite produced by plants and is generally very bitter to insects. It is said to be a CNS, that is central nervous system stimulant, which helps us to get more attention. So in our current work that we aim to establish the relationship between these things, like how diet affects the architecture of Drosophila sleep and wake behavior. So our main objective was to study how if we change the diet of the Drosophila, would it affect their behavior, and then how caffeine could affect the sleep-wake cycle in them. Next. So the methodology which we used is first we took the culturing bottles, and then we had put banana slices in them as well as the banana peel. Then we have kept them for about 2 days so that we would collect the cells. And then after 2 to 3 days, we took up those bottles, cotton plastic, then brought it back to a lab. Then we took a normal, that is the control media, that is the Rawa jaggery media. Then we transferred the slice and we subcultured them. And then we sterilized the First is that of a controlled media which had normal Ramadani media, whereas caffeine media had a caffeine addition. Sucrose, agar sucrose media only had agar and sucrose. Agar sucrose yeast media had three of the components. So these are the various dietary modulations that we did when we cultured the flies. So this is how we monitored the flies for identifying how a Drosophila is sleeping, how it is sleeping. We just took the theoretical knowledge that prolonged periods of immobility, if they are not moving for a certain period of time, they are taken as proxy for sleep. So we noted about the videos were run for about whole day and night for 3 to 6 days, and then the videos were analyzed for how they behaved. Next. So these are the results that we got. First, in the caffeine media, flies showed continuous activity. They had continuous activity as compared to that of the controlled bottles, where they were immobile for some time. And it compared to the controlled flies, it go into stable state. And it showed moderate social interactions. Whereas the AS media, that is the agar sucrose media, the flies showed highest locomotor activity. By locomotor activity, we devised that they had a fluttering speed when we saw it in the bottles. Whereas compared to the control flies, where they were very little and they didn't have a very fast movement. So they also had a very good interaction for a couple of days, and they had a small nap test. That is the rebound state that I was talking about. When these flies don't have enough sleep time in the night, they take some part of the day, some circadian time, and they take afternoon naps. The AS5 media, that is the agar sucrose media, the flies are behaviorally moderate. There was no big change, but they do have a nap time, and they sometimes showed particular patterns of social interactions. Next. So these are the certain captures of the social interaction. When we see the day one captures, we mostly see the social interaction captures, we mostly see here these dots are the flies which are isolated. They are all solitary accordingly on the day one. But when we see the flies on the day 4, this is the AS media example, that is the agar sucrose media. We can see these two flies are being together, but there are some certain interactions that have increased. Next. This is the day 5, that is almost last to the day of fly interactions. This is all the 3 media that we have prepared. This is AS5, and then you can see 2 flies are interacting here, and here's 2 flies that they are interacting on the media, and here 2 flies have interacting in the caffeine media. So according to theoretical studies, it is said that mostly these flies are solitary, but when we saw in our dietary modulation, they were interacting more, and the different patterns were like 2 to 3 flies were seen together, or they were. It is said that this kind of groupings is said mostly for mating or foraging activities. So we concluded that caffeine increase the wakefulness times in flies, and sucrose diet regulation increase the locomotor activity. That is the flirting action that we saw. It was noted mostly that according to the previous study, that the solitary action, they were known for their solitary actions. But after we made certain changes in their diet, we saw that there were some behavioral changes and they had more social interactions. So that is what we concluded. But we had certain limitations in the study. As it was a basic study, we didn't have enough of the current technologies that would help us get clearer observations, like Drosophila monitoring system, ATPSAs. So the study lacked in certain bit of the technical aspects. These are the few references that we used. Thank you.
Thank you. The session is open for discussion. If anyone have any questions, you can ask. Yeah, okay, thank you. Next, welcome Priyadarshini and team, Katham Academy of Higher Education, to present their paperwork.
Good evening to all. Myself, Rita Manager. Myself is Sri. Today we are here to present a project water quality in biological lakes. As we all know, water is an important component for living and it is a major part of ecosystem. There are many types of aquatic ecosystems like oceans, ponds, swamps, backwaters, and lagoons. And Meta is one of the major aquatic ecosystems. Meta has a defined and a unique ecosystem and also it plays a major role in maintaining and researching groundwater and also treating groundwater. So we call it as a piece of earth. As we speak of water, it is a brief and great history of wetlands because it dates back to 8th and 9th century of Cholas, where they created them for irrigation and river management purposes. In the past decade, we all know Coimbatore faces a rapid urbanization and industrialization due to which the lake waters are depleted and degraded rapidly. So, we aim to check the water quality by physiochemical parameters to ensure the safety of water which is being used by humans and animals. The main objective of our project to determine the steady area by review article, collect the data about steady area, collection of samples from lakes, analysis of physiochemical parameters.
Let's start following methodology. Study area. Study area. Without choosing the study area, point to lakes. Next, after the study area, after selecting the study area, collecting the sample, collecting data collection from previous articles. After data collection to sample collection like lake water. After sample collection to assessment of the water quality in various parameters like BOD, PO, pH, alkalinity, hardness, chloride, and phosphate, nitrate, COD, sulfate, TDS. These various parameters to restrict to obtaining this result, comparing the result with Indian water quality index. The sample collection. We are choosing the verified lakes from Coimbatore. First one Sulu, Singanallur, second Balagulam, Chinnakulam, Periyakulam, Kurichi, Kumaraswamy, Krishnapalli, Vedapalli, Peru. This picture is ground collection in study area. Chinnakulam, next Balagulam, Singanallur. Kurichi, Peru, Vedapalli, Krishnapalli, Kumaraswamy.
Materials and methods. First, we are collect the sample and take to laboratory and analyze immediately. The pH was measured using pH meter. The BOD and BO calculated Winkler titrating method. Sample for 5 days incubation at 20 degrees Celsius. COD estimating the potassium dichromate presence of strong acid and titrate against ferrous sulfate using ferrite as indicator. Sulfate concentration was determined by turbidity method, chloride by archinotometric method, alkalinity was determined by titrating method, the temperature was determined by thermometer on the spot. TDS will analyze laboratory and hardness analyzed by using EDTA titration method. Now discussion and result section take over the main session. As we speak of results, we compare the results with WHO guidelines, Indian Water Quality Index, and EPA guidelines. First, pH. When we see a pH, all the values comes under these guidelines. And when we speak of temperature, again, it comes under guidelines. In chloride, there is a elevated level in 660 and all other lakes comes under the EPA guidelines. As we speak of hardness, again, Singanallur has highest value and Kumaraswamy, Peru and Vedapalli was elevated levels. And in alkalinity,
In PDS, Chinnakulam is very much elevated and all other lakes comes under the standards. All the lakes has a normal phosphate level and all comes under the guidelines. In nitrate, Chinnakulam has comparatively very high level of nitrate and all other lakes comes under the guidelines. As we speak of DO, every lake is very much polluted and it is very low. So the value of DO is not up to the standards. Then BOD, as we speak of BOD, every lake is polluted and it does not come under the guidelines. When we speak of COD, all lakes except Vellore and Sulu comes under the guidelines. And finally, sulfate, in Simhanadu lake has an elevated level and all other lakes comes under the guidelines. So this is the Indian water quality index to which we have compared our results and concluded. So the conclusion is that each and every lake has its own elevated levels of different parameters, but in the end, Simhanadu, Vellore, and Chinnakulam is very much polluted compared to other lakes. So there is a need to analyze and monitor the water quality in a regular basis to ensure the safety for humans and animals when they use these waters.
Thank you. We may not have any questions.
What are the apart from these parameters, you have concluded that it's highly polluted. So do you find any other species, any other indicators like microbial indicators that you have checked for? Actually, we are working on freshwater biodiversity fishes now. When we speak to fishermen of these lakes, they told us that there is no natural varieties in these lakes except Oleograms mosaicus, the local name jellyfish. All other fishes are artificial. They are getting inputs from the fishermen and they are rearing it in the ponds. They say us that there is an elevated level of pollution. Due to that, there is a depletion and degradation in the biodiversity of fishes. So we can also check the pollution levels by the fishes. And also we can check it by DNA extraction and analysis of heavy metals in the fish. Do you find any metal? Actually, we have given our samples for IPCS analysis. It is so we. That phenomenon is the most regulated level because it's very much polluted with no, you have taken only 10 lakes.
Thank you, team, for your presentation. Next, I call upon Ms. Vidya from second year MSc, Alpha Academy of Higher Education, to present her presentation.
Good evening to all. I am Divya from second year MSc Biotechnology. This is my MSc thesis, which I am going to under topic of morphological and osteological characterization of the genus Epiphyllus of Cyprinidae family and Oleocomus mosaicus of Cyprinidae family. introduction. Nowadays, water is a crucial survival of all living. It's the essential. Water is the essential of all living, except for us, humans, fish, animals, birds, etc. And in the fresh water, the ecosystem is Earth aquatic ecosystem. Water in the two ways, fresh water and in marine water. In fresh water, it's I have collected the samples from Royal River. It's a Royal River is a branch of tributary Carbeti, and Royal River flows through the four districts in Tamil Nadu, Coimbatore, Tirupur, Erode, and Kannur. The Royal River is originated from the Nilgiri rocks in the western Ghats, the western side of Tamil Nadu. The latitude of Royal River is 11 degree 2 seconds north, 11 degree 30 minutes north, and the longitude is 76 degree 30 seconds east and 78 degree 30 minutes east. Freshwater and fish. There were many fishes, there were many types of fishes in freshwater. And then around currently there are 10,000 different types of fish they have there. Considered. In that, the fish have a small to big size. Where by the testing, it's a comfort under 3 centimeter to 2 meter. Heavy metals. Heavy metals is a water polluter, water bodies are converted into water pollution. Why I'm taking this means fish is the first indicator of the climate change in the water. Fish is the first indicator. While climate change and the heavy metals, the fish is observed that it can automatically adapt to the condition. So that fish is first indicator. In that, while a fish consuming the food in under the water, it will consume heavy metals also. In that, large concentration of the heavy metals are stored in the gills. And in the fish organs, gutters are absorbing metals from the sediments. Sample collection. We have sample collected from the Coimbatore and the branches of Royal River. We have chosen the 3 places: Chitrai, Chavadi, Kanapalai, and Tenkara. In that, there are many species of collected. I have to take 2 species: Divaria acumentus and Oreochromis. Morphology. Morphology is the external and internal structure of the morphology is the external and internal structure of the fish. Fish is not all living organisms. And morphological characters of the fish are identified by using a diagram, which I have taken as diagram 1999 and 2010 is a book. And they have released 2 books, the year of 1999 and 2010, at this website. The fish characters are identified and measured using vernier. Osteology is a scientific study of bones. It is the structure of bones and skeletal elements, teeth, pathological, and etc. Method which are chosen as a Taylor and Manning method. Till this day, this method is followed for osteological method. For morphology, I have taken 1 specimen for 2 species also. For an osteological study, I have taken 5 specimens for each species. And then for osteological, I have there are 2 staining methods of doing. One is cartilage staining and bone staining. Cartilage staining is the blue color image. Blue color image is the cartilage staining, which are used as Alcian blue. For the visibility of individual cartilage in fish. And bone staining is for dorsal fin. There are 4 types of fin in fish: dorsal fin, anal fin, pectoral, and pelvic. to visibility of the fields and trees. Results in morphological there are 24 morphological characters and 20 meristematic characters. This is the external structure. Dorsal fin, anal fin, pectoral and pelvic. These are the measurements I have taken for two species. One is Divaria acupenatus and species two is Moriodomus mosanicus.
Species one is Divaria acupenatus and species two is Moriodomus mosanicus. First, I have taken the total length of the species from the mouth to cattle fin, and second is the standard length. Likewise, each character was measured. These are the meristematic characters. There are 20 characters which I have taken. Osteological results. The osteological results I have observed a fish for 1 month under the staging. These are the measurements which I have taken for 5 specimens for each species. These are the meristematic characters. In osteological, in fish, there are 3 types of fins. There are dorsal fin, anal fin, which is supporting to our body. Caudal fin is the main organ of the fish. Dorsal fin. Dorsal fin are in 2 ways. One is a stronger and smooth spine, and another one is weak and smooth spine. In that, I have observed Oreochromis mosanicus and Divario. In Oreochromis, the spine was which is stronger and smooth. In Divario, the spine was which is smooth, weak and smooth. These are the skeletal structure of the dorsal fin. Anal fin. Anal fin is a part of a pattern of a dorsal fin. Anal fin is present less than a dorsal fin. These are the structure of anal fin. Caudal fin. Caudal fin is the support of 3 caudal vertebrates in the axial skeleton. Basically, in the fish, there are 5 to 7 hyperioles are present. In Divario, 6 hyperioles and 1 perineal are present. In that, 5th and 6th hyperioles are very small. In Oreochromis, 5 hyperioles and 1 perineal are present. In that, 5th hyperioles is very small. Any other questions? Oreochromis for the lateral scale is around 36. Divario for the 45. அங்க வந்து நிறைய fish கிடைச்சிருக்கு. பட் இந்த 2 ஸ்பீஷிஸ் தான் லாஸ்ட் நம்பர் ஆஃப் varieties. ஏன்னா ஆஸ்ட்ராலஜி அந்த மாதிரி பாத்தீங்கன்னா நிறைய ஸ்பெசிமென் வேணும் நமக்கு. ஏன்னா ஒவ்வொரு வந்து ஒவ்வொரு ஸ்டேஜ்ல டிஸ்ட்ராய் ஆயிடும். இப்போ நான் பண்ணதே பாத்தீங்கன்னா 5 ஸ்பீஷிஸ் பண்ணேன். 5 ஸ்பெசிமென் இருக்கு. அதுல வந்து இப்போ 3 தான் எக்கிட்ட இருந்துச்சு. டிவாரியோல வந்து 5 இருந்துச்சு. ஓரியோக்ரோமிஸ்ல 2 தான் இருந்துச்சு. ஏன்னா அது ஒவ்வொரு ஸ்டேஜ் அது மாறும்போது அதோட pH அந்த மாறும்போது அது வந்து டிஸ்ட்ராய் ஆயிடும்.
Yeah, thank you. for this presentation. We are remaining. Yeah, okay. Thank you all for this oral presentation. Now it's time for poster presentation. I request our presenters to post.